I know, I usually do too, and I just.
The one time you know is like actually a question.
Well, actually, I find the problem is the fact that it's the version that's, it's the shared version that goes, you have to, my version, I have to access, see if it's an internet to use it on a system computer. So it's like, even to open files, it's like, ooh, it opens when it wants to open.
You talking about your Microsoft Word? You have to get on St. Vincent's like internet and go through them to get on that.
Yep. Yep. So I open it up, and right now it's doing loading add-ins, 1 of 1. I don't like it at all. Having the control of my stuff, not in my hands, makes me angry. I become, I horrify. Please. Definitely true. So you and Emily can keep talking about stuff.
It's also relating to this class too.
It really overlaps a lot.
It does.
At least this the first unit for finance. Before
BRB. Be right back.
yeah. That comes back a lot. Yeah.
The question was like, yeah, they are exactly the same. It was like the last one I had. It said like 98.
Serotonin.
The last.
I got one.
Oh, I know. Remember.
On here today.
How much one is supposed to be?
So speedy. My office is right here on the hall. Father, Son, Holy Spirit. God, grant me the serenity to accept the things I cannot change. Courage to change the things I can. Wisdom to know the difference. Father, Son. All right. Yes. So today, we're going to talk about, we were going to talk about acetylcholine, not doing it. What's that?
Nothing matters.
Were you ready to?
I was ready to, but you don't need to. That's all right.
No.
Another day.
We'll start that next week. So, norepinephrine, once again, when we're talking about this sort of system, we use the term adrenergic, right? Adrenergic. Adrenalinergic. And we talked about two major pathways. One is that locus coeruleus, shown here, originates in the telencephalon. Excuse me, or excuse me, the pons here, the pons. And then syntaxons, lots of areas. Almost every area of the forebrain, so the forebrain are all of these areas of. And, so to get to your question, there is reciprocal innervation between, so the locus coeruleus sends, and this is just for your information, so this is not like, you don't have to remember this. So the locus coeruleus sends neurons up to the amygdala, to a region called the basolateral amygdala. Now, this is the detail that, even though I read the paper I read it real quickly on Friday, I don't have all the details. So the central amygdala sends neurons back to the locus coeruleus. Okay.
What's the
Central, is that central amygdala? And this is basolateral amygdala. So yeah, so the amygdala, oftentimes you'll just hear the amygdala spoken about, and here's the amygdala. But like a lot of regions, it's actually broken up into little sub-regions. So it's not just like one area. But anyways, and then we've got this other whole big system, adrenergic system, that affects our periphery. Lots of our major organs receive norepinephrine as a neurotransmitter, and norepinephrine as well as adrenaline or epinephrine are released from the adrenal medulla as a hormone in the blood, where it has effects all through the body. Okay, we talked about the receptors, right? We did talk about these. Then we talked about I'm a little frazzled here because the computer sort of gives me problems. All right. So we stopped on Thursday talking about wakefulness or arousal. Right? So wakefulness, staying awake. Arousal means attention. Like, whoa, you got my attention. That's arousal. And we were using the results from this experiment to give us, it was a little data interpretation exercise for us. And I think what we found was that if we activate alpha-1 adrenergic receptors in the hypothalamus, what happens to wakefulness? Does it go up? It went up, right? Just look at the data. You know, here we don't want you to look at your notes, just look at the data, right? So wakefulness is represented by the seconds awake at different time points after infusing these drugs into the hypothalamus. And what happened when we infused that? Phenylephrine looks just like epinephrine. What happened to wakefulness? Okay, so it wakes you up. What about activating beta-1 adrenergic receptors? Very good. Now, these drugs, and not only did they increase it, but they increased it in what's called a dose-dependent manner. Dose-dependent manner. That's really important for pharmacology. That's one of the key, and we're definitely going to talk about that. We're going to come back to that a lot once we really start diving into the pharmacology, pharmacokinetics, the properties of drugs themselves. So when you see a drug, when you're looking at a paper, and you see, oh, this effect happens in a dose-dependent manner, that's one of the clues that tells me that this effect that I'm seeing is due to the drug effect, not some other thing. Yep.
So does the caramel have similar like effects to the phenylephrine? Like does it like constrict the blood vessels and reduce excretions, or is it like just like this?
Okay, that's a good question. In your body, so we can do a little physiology review here. So first of all, activate alpha-1. Boom. What happens? What do we get?
Increased norepinephrine.
Well, that's already happened. So norepinephrine is released. Binding to alpha-1, I get.
Blood vessels constrict and it reduces secretion.
That's true. What's one of the reasons that it does that?
It increases arousal.
Well, so we activate these receptors and then it has some sort of effect.
It involves calcium.
Yes. As a second messenger, good. Then Emily, you're right. Then Emily, you're right. You get, with blood vessels, we get constriction of blood vessels. In the brain, if we're acting on other neurons, this is probably going to activate or excite neurons. But in the body, we said that constricts blood vessels. Neurons, we're going to say this is going to activate neurons. This should be excitatory, right? I'm pretty sure we did. Didn't we say excitatory, inhibitory? Did we do that? I'm just making sure. Okay, good. Well, we talked about, so phenylephrine was in general, or that was a specific drug effect let you know, it also acts as a decongestant. But in the brain, it's activating these alpha ones, which means that it's having a norepinephrine-like effect in the brain. And then sort of to link that with what might be happening in neurons, you got to go back and see what we talked about with alpha one. So I can see where where that got lost. That makes sense. So in the brain, when we're talking about what norepinephrine does, it's not about blood vessels. It's about activating or exciting neurons. Okay. Alpha 2 is something else. It's an autoreceptor. Right? Alpha 2, what do autoreceptors do? They inhibit the release of the neurotransmitter. Beta 1. Increase in CAMP, which is excitatory. So in neurons, this is going to excite the neurons. If this comes in and hits your heart, increases heart rate. This comes in, different effects. It depends on where the receptor actually is. And that actually brings up a good point, because if a drug activates this system, let's say you give a drug and you're trying to you give a drug and you get both of these effects. You see here, it's activating alpha-1 receptors, leading to increased wakefulness in the brain. This is brain, remember this is brain. Or if you activate beta-1 receptors and I increase wakefulness in the brain, what kind of other effects in the body would it produce? It's going to have effects, we're going to say, all through the body. You might see constriction of blood vessels.
But you had a decrease in digestive activity.
You could see constipation. You could see nausea. Nausea, constipation, vomiting, and diarrhea. Those are like 4 common side effects. It's either coming or going, you know. It's either go or no go, or get it out of your guts, right? So those 4, so the guts are always going to be affected. Or it's a common side effect when you start affecting these systems. You give a pill, it's going to hit all the receptors in your peripheral nervous system too. It doesn't just go up in your brain. Okay. We might also see heart rate increase. Right? So just something to think about. I'm not going to ask you this stuff. So in general, these are just some data that tell us that if you increase adrenergic tone, or if you activate these adrenergic receptors in the brain, you're going to increase wakefulness. They both contribute to something. Okay. What about if we activate alpha-2 receptors? So alpha-2 receptors, they're going to oppose the alpha-1. Or actually, alpha-2 is going to just reduce the release of norepinephrine in general. Right? Because they are autoreceptors. So, now, we saw the monkey completely kick butt on this task on Thursday. So I don't know where they're getting these data. If they're distracted, their efficiency goes down 5%. I wasn't seeing that in that monkey, but whatever. So what they did was, the monkeys observed the experimenter placing the food treat in one of two wells. And the wells are hidden from the monkey by a screen for a period of time, and that can increase or decrease. That's their way of trying to distract the monkey. Then it has to choose or remember the correct well to receive the treat. And sometimes an additional distractor stimulus is introduced, lights or bells or things to try to keep the monkey from remembering where the treat was. So what they saw was, here's our control, here's the performance of our monkeys under control conditions. You distract them. Again, I don't know if I'm believing this. So these are the data they collected. So what happens? What happens to their ability to remember where the treat was? Good. A lot went down. Thank you. Good. That's pretty basic, right? The red line is lower than the blue line, so impaired memory here. Then we give the guanfacine, an alpha-2 agonist. So remember, when you see that, what's that doing? It's probably reducing the release of norepinephrine by this adrenergic neurons. So we can see release of reduced release. So it's going to be at these axon terminals, reducing the release of norepinephrine. Thalamus, hypothalamus, amygdala, cortex, etc. What happens to their learning or their memory when you block, when you activate this alpha-2 receptor? Compared to distracted state without. What happens to their performance?
It's slightly better.
Slightly better. It's actually better than it was at baseline. So do you see that, first of all? Do you guys notice that? Yeah. Okay, good. It increases. Allison, are you with us?
Yes.
Okay. I got your head on. Touch that screen. All right, good. So what's that tell me? What is that telling us about adrenergic tone in general? About norepinephrine levels in the brain? I'll tell you what. I want you guys to take a look at this graph for a few minutes. And I want you to interpret that for me based on everything. So let's break down what we've said so far. Okay? Let's write the data down. from our two little experiments, let's pull these together. Number one, I activate, we're going to say alpha 1 receptor. What is that in a sense imitating or substituting for? If I activate alpha 1, is that a norepinephrine-like effect? Yes. So what do? I'm increasing adrenergic tone. We did talk about that concept. Dopaminergic, serotonin. No, you guys remember that, right? So, okay, I'm over my stress. Are you guys like that? Is this like a bad week for you guys? Okay, I'm just checking because you guys are just like super quiet. All right. Okay, so let's go to number 2. Alpha 2. Oh, let me put my little drug in here. Phenylephrine. 1-15. Okay, I'm activating alpha 2 now. What's that doing to adrenergic tone in the brain? Allison.
Inhibits.
Okay, good. You're quiet. Try it now.
I said it inhibits because they're autoreceptors.
Good. I just had to increase the L. Okay. All right. So what effect did number 1 have on wakefulness? Improved it. What effect did number 2 have on cognition? On cognition. On cognition. Knowing what's happening. Boom. What happened to cognition? Improve. Improve. Right? Okay. Now you got all the tools you need to interpret what's going on in this graph. I want to hear what you guys have to say about that. So what I want you to do is take that information, and then I want you to take this figure, and I want you to summarize it for me in 2 or 3 sentences. Go. That's for you too, Allison. Did you hear that assignment?
Yes.
Okay. Yep. So summarize this graph. Let's write some notes down. This would be a great thing to do on an exam question too.
I agree, but.
Yes, what do you think, Al? Woo-hoo, woo-hoo. I think too. This is what you guys get when the computer crashes. This is actually a good thing, so think about this. I want to add in one additional piece of information for you. In the peripheral nervous system, when I increase norepinephrine release, no, I'm going to say this, increase adrenergic tone this is typical of what kind of response to a stimulus? Blank or blank? Okay. Fight or flight should be otherwise interpreted as I might die right now. It's probably the best way of anthropomorphizing the fight or flight response. Let's throw all that in the bucket. For you to think about.
Every day.
Oh,
Or not.
I was sad it was a great 3 day, about 30 minutes ago. Okay, you guys have thought about this for a few minutes. What do you think? What's this graph? What is this summarizing for me? What's this telling me? Let's have some thoughts here. Okay, we're going to go all around. That includes you, Allison. We're going to let you chime in here. Okay, so let's start us off. Julie.
And here are my thoughts.
Mhm.
Okay. You love him first?
Just the way you said it was funny.
And here are my thoughts.
Yeah.
With the increased activation of alpha-1 receptors, which will increase the adrenergic tone, too much of that activation of like adrenergic tone is going to create the higher fight or flight response, and that higher level of stress will decrease the working memory performance, and kind of like the opposite for the alpha-2 receptors.
Okay. So we need some. Okay, so let's tag on to that, Emily.
I said that the optimal working memory performance is a balance between alpha-1 and alpha-2 receptors in order to kind of, maintain not having high stress or low, I guess it would be low sedation. So in order to have that good working memory, you need to have balance between them.
Good, I like this. Devin.
All right, so I think it is a scale of adrenergic tone. So low is low amounts of the hormone, and high is whenever you get too high, there is now a lot less cognition factors because the alpha-1 pathway is activated more, and there is nothing but wakefulness, and now you might die. But if you had too little of this in your system, too little adrenergic tone, you would be in your state of state because you would not be wakeful at all because there's no activation of the alpha-1 pathway.
Good, I like that. Very good.
Everyone is also looking at it. So I think what I can really add is that a too high adrenergic response is going to significantly impair cognitive capabilities because I guess the sympathetic response would very much be about like great responses, and that would be very much what the word, like reflex-like responses. Not you don't have time to think, which monkeys were a little bit different, but it's still be a similar response for them. And memory is a cognitive, like memory would be impaired by high stress form. And obviously, the A2 receptor, alpha-2 receptors are too highly activated, and the monkey basically studied it completely and also significantly impressed cognitive capability.
Good. Allison.
So I just said that when the alpha-1 receptor is activated, an increase in adrenergic tone helps improve wakefulness, and adrenergic tone is necessary for a normal level of wakefulness. And that's related to your fight or flight response. And then as alpha 2 receptors are activated, cognition's improved, and there's an inhibitory effect on wakefulness.
Okay. So the one thing I would say is that when alpha 2's activated, so we have the balance. We're in the middle here. It says optimal. We're in the middle. We are awake, and we are able to pay attention to what's happening. Like in the classroom, we can pay attention to what's happening. When we're reading a book, you're paying attention to what you're reading. You're attentive, and you're actually retaining that, synthesizing that. So we need a certain amount of alpha 1 activation in the brain for that. You get too much? What's the extreme in animals? What's the extreme stress response? That is very extreme. I'm thinking one step before that.
Fear.
And what's a fear response in animals? Rodents do this all the time. The freezing response, right? So the freezing response. So you're freezing. Are you paying attention to what's happening? You absolutely are. Are you thinking about anything else? No. So when you're way out of balance, you're aware, but you're only aware of one thing. How am I going to survive these next 15 minutes or whatever? That's the only thing in your mind. That's why when you're stressed out, it's really hard to study, or take an exam, or remember anything. Because your alpha 1 receptors, they are just getting blasted. Now, anyways, so we're going to see this with serotonin also. See this with most of these systems, is that there's an optimal range wherein this is the norm, and if you get outside of those norms, you're going to have your extremes. So people with depression, for example, one of the reasons that they are depressed could be maybe they just have either too few alpha 1 receptors, too many alpha 2 receptors, or maybe their neurons don't make enough of the neurotransmitter. There's lots of different. Hey.
Were you saying that too much too much adrenergic tone, so too much wakefulness causes stress?
Yo, that's right. Too much.
Too much.
It goes from I'm paying attention to you to that.
Like fixated.
Well, I think it's more intense than fixated. Like it is a life or death situation.
It's like whenever you exam, you start reading because you don't know the answer for like immediately.
Oh, yeah.
And you can't think about it anymore.
Okay, Allison, let's try that. So let's say it's Thursday. Oh wait, that's 2 days from today. I'm being a bully right now. Yeah, that's good. You smiled. That was like a partial smile, like, okay. So, yeah, so you've had this experience where you flip to a page on a test and all of a sudden you're like, oh my gosh, I know this, but it's all gone. Now how do you feel? You feel stressed. And then the more you think about it, the less you can remember. Why? Because you're releasing all your stress hormones. Because our brains don't know the difference between I'm going to die or I just don't know this material for the test. That's why it's good to listen to some relaxing music or things like that while you're studying. Lavender can be helpful. It's very relaxing. Lavender. Anyways, so, cool. Other questions out there, Allison?
No, I don't have any more.
Okay. So does that get you? Does
Yeah,
help
that
you out?
makes sense.
Okay.
I
So
think
you had
that
these
the general idea, like the takeaway, is that there has to be a balance level. There can't be too much and there can't be not enough, because that's when things start to not function properly.
That's correct. And drugs that can help people with, like if you give an adrenergic, an alpha-1 agonist kind of drug, if you were to give a drug that, actually, those don't exist. But let's just say you were to give a drug that helped with learning. Well, let's say the guanfacine, okay? We see the improvement. Now, this drug is going to probably be helpful for people who are out on this range of adrenergic activity normally, right? Why? Julie?
Hopefully bring them back.
Bringing them back to balance. What if it just puts them to sleep? You either gave them too much drug or they're already at their optimal spot. Okay, good. Okay, so these are things to think about. Cool. Serotonin, this one is crazy. We're going to move Allison over here. See your face, but not all over the slide. Okay. Serotonin starts as the amino acid tryptophan. Turkey, turkey, turkey. Thank you, thank you. Serotonin was actually first discovered in the blood. It was first discovered circulating in your bloodstream, not your bloodstream, but the bloodstream. Erase this board here. It was discovered circulating in the bloodstream. Right. And its effect was to increase constriction of blood vessels. Let's fix something. No, I lost it. So, bloodstream. Zero. Constriction. Tone. This molecule increases the tone of blood vessels. Therefore, we will call it serotonin. Isn't that cool? And it's synthesized from gobble, 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 amino acid, the Thanksgiving amino acid, which we will be able to enjoy and not have to come back and take exams this year. That's a drug-free day. Anyway,
What we do have exams afterwards.
after Thanksgiving,
Online finals and after.
wow. I thought finals were before Thanksgiving. Oh, that's right, sometimes they are online. Okay.
Some finals are.
Sorry, wrong.
Sorry
Where? Okay.
for bringing you guys on Thanksgiving, you know, acceptance.
So it's tryptophan is this enzyme tryptophan hydroxylase adds a hydroxyl group to the tryptophan, and then aromatic aluminum converts this. It takes the carboxyl group away, decarboxylase. So this is the final form, 5-hydroxytryptamine, or 5-HT. The major neuronal pathways. Okay, so you know synthesis, you know that we've done a lot of synthesis pathways. So you should know the precursors for these things. What's the main one for dopamine and norepinephrine? Well, take a look at that. Okay. Later. Okay, so serotonergic neurons. They originate in the pons in the medulla in clusters called the Raphe nuclei. And just by the way, these letters, so they used a particular marker to identify serotonin neurons. And so these ones are all labeled Bs. And if you might remember, the ones for the catecholaminergic neurons, those were all labeled with As. Same thing, they just labeled those clusters. Now there's a lot of clusters here. We are only going to focus on B7 and B8. These two here. B7 is the dorsal raphe. B8 is the median raphe. So B7 and B8, that's what we're really going to be looking at. And what's important, the reason that we focus on these two regions, I know it looks like it's coming from B9, but B8 sort of swings around this way. So look at all the areas that are innervated by these two regions. We've got the thalamus, the hypothalamus. That's B7. B8 comes out here. Thalamus, hypothalamus. Uh-oh, there's our buddy, amygdala. Globus pallidus, caudate putamen, part of the basal ganglia, hippocampus, amygdala, and these are hippocampus, amygdala, part of the limbic system. Yo. Then we have the frontal cortex up here. So these are pretty important. Now, there's tons of serotonin, kinds of serotonin receptors, but let's look for a moment in the brain. Serotonin neurons, they terminate on dendrites rather than on cell blocks, okay? Now, one of the reasons that we know that is some evidence given here in this microscopy picture. So this is a slice of the brain. This is the hippocampus. By the way, is this not like one of the most beautiful, isn't that beautiful, hippocampus? I love the hippocampus. It's a beautiful shape. Love it. But anyways, so this is what's called dark field microscopy. You don't need to know that, but dark field microscopy, basically what it says is that when the structure is stained, it appears white on the picture as opposed to dark. So a lot of times we've seen things stained where the thing that's stained is dark or green or red. With dark field microscopy, what's actually stained is white. So one of the reasons that we know that serotonergic neurons terminate on dendrites is because this region here, this is the outermost layer of the cortex that contains dendrites of pyramidal neurons in the cortex. not soma. And this darkly stained region here in the hippocampus contains dendrites of neurons in the hippocampus rather than cell lines. Okay, that's just a little FYI. Man, there are 14 different serotonin receptor subtypes. You need to know them all. No, I'm just kidding. So we are going to focus on these three kinds predominantly. So notice. 5-HT1, and then there's subclasses, which means that all 5 of these, for example, A, B, C, D, and F, they all function very similarly in the same way. So when these receptors are activated, they're going to have a similar effect on the receptor and on the neuron. What distinguishes these would be the affinity that the different classes have for serotonin. So serotonin might bind more highly to the 5-1A than the 5-1C, for example. All right. Okay. Just to show you those. But we're going to focus on 3 compounds. 5-HT1A. Remember, 5-HT is the serotonin. Okay, so these are found in projection regions for the serotonin neurons. So when those neurons are reaching out, cool. Yes, shoot. This is actually, I want to spin another slide here. This is another big thing here I want to point out. The 5-HT3. These are ionotropic receptors. These are ionotropic receptors. So when you hear that, let's see how your adrenergic tone is. Is it way over in the alpha 1? Is it way down in the alpha 2? Or is it like right in the good spot? Ionotropic, what do you think of when you hear that term? Sedation?
Yeah.
Sedation mean what? So I activate the receptor and what happens?
The activation of alpha 2.
Oh, no. Oh, okay. Now I'm getting back to it. I'm trying to see if you remember. That was my goofy way of trying to see if you remember. What does an ionotropic receptor do? What's happening to your heart rate right now? Is it going up?
Yeah.
Okay, so we're hitting alpha 1 a little too much.
Allows
Oh, hello.
just allows ions to move back and forth.
Well, these are ion channels. So the 5-HT3, these are ion channels. The rest of these are those crazy G protein coupled receptors. So 5-HT3, of all the different kinds of serotonin receptors, the 5-HT3 are the only ionotropic ones. All the other ones are G protein coupled. Right? That's important. Forgot to mention that. Okay. Serve by activate 5-HT1A. 5-HT1A. I'm going to activate a G protein. The first thing I'm going to do is I'm going to activate a G protein, which leads to a decrease in cyclic AMP and increased potassium efflux. So this has an inhibitory effect on that neuron. Right. HT2A. Activated G protein. Activated G protein, which is going to lead to calcium influx. Okay. Now, 5-HT3. These receptors are actually on sensory input areas of the vagus nerve. It sends information back up to the brain to let the brain know what's happening. You activate that, 5-HT3. This opens the ion channel. We get an influx of cations. If there's too much, you puke. Ends up activating the area in the brainstem called the area postrema. Ralph, hurl, puke, vomit.
Is that in the myencephalon?
You should check that out. Upchuck. Hurl.
Is it like, I was in like a class in high school and some kid was like lifting too much weight and then like he like passed out and then like threw up because of his, to his vagus nerve.
Probably, yeah.
Is that right, like you said?
Yeah.
And he got choked and really so bad.
So when your alcohol levels hit too high, too much alcohol in your stomach, body says too much, too much. Puke, hurl, vomit, yak, blats, blow chunks. Anyway, hey, let's move on from there. But this is actually, this receptor subtype, we're going to talk about this. This is actually used by so emesis is the actual technical term. Emesis, E-M-E-S-I-S. You don't need to know that, but that's the technical term for vomiting. And antiemetic drugs prevent that reflex. Physicians and Pharmacologists develop drugs that block these receptors to prevent chemotherapy-induced vomiting and nausea, to help patients tolerate the drugs, and whether that's a good thing or a bad thing, that's a conversation for another day. Okay. Let's do a quick round the world drug tour. Buspirone. This drug hits lots of different areas. It's lots of different kinds of receptors. Buspirone is called, it's like a dirty drug because it hits all different kinds of receptors. But one of the places that it does do, is it activates 5-HT1A receptors. So buspirone activates these. The example that the book gave. Now, pay attention, right? Serotonin's backwards compared to all the other ones. All the other groups that we've talked about, dopamine, norepinephrine, well, yeah. The ones are the activators, the twos are the inhibitors, right? In this case, it's flipped. Serotonin. Why? Because they found the 5-HT1 receptors first. So it's flipped, right? I activate 5-HT1, I inhibit. Just remember that. It's going to increase anxiety. Decrease anxiety. My bad. Thank you. Okay, Devin, this one's for you, buddy. 5-HT2A LSD. You know what that does, or maybe you don't, but we're familiar with what that does. We've read about it in books. Hallucinations. So it can help increase your sensory experience, but if it increases it too much, that's when the walls start to talk to you, right? They're blue, man. It's like singing to me right now.
Then that's not a drug-free day.
That's not a drug-free day. Now this Ritin, I just am throwing this in here, Ritin sarin. I'm probably saying that incorrectly. Ritin sarin. This is used more for in studies, animal studies. It blocks 5-HT2A receptors. So one of the things that they will do with these drugs is, let's say you give a drug. You give drug XYZ, 1, 2, 3, 4. And the animal is like exhibiting some sort of effect. Well, you know it's hitting serotonin receptors in some way. You give this drug, which blocks the 5-HT2A receptor, and now you give, well, let's say we'll give a dose of our LSD here, and now the behavior in the animal that you saw with LSD alone goes away. That tells me that LSD is exhibiting its effects by activating these receptors. So this is just a little intro into some of the tools that we can use to help us identify what receptors are causing the effects.
So they just basically cancel each other out.
That's right. That's right. When it's bound, so when this drug is bound to the receptor, it prevents the LSD from binding. Okay, good question. I'm just introducing that concept here. I just wanted to bring it in. And then here we block the 5-HT3 receptors. And I already told you that's an antiemetic, so 5-HT3. On Dancitron, that's a this is a 5-HT3 antagonist, blocker. I mean, it's basically what the word antagonist. A drug that activates the receptor we call an agonist. So this is opposed to activation. Simple, okay? What you do? Decrease chemo-induced emesis, vomiting. So it's going to block those 5-HT3 receptors that are on the vagus nerve. All right. Now I think this is pretty cool. So who's heard of the guy called Jordan Peterson? Anyone? Okay, this is an anti-Jordan Peterson slide. Not anti, not against them. Try to. Come on, Allison. How can I'm going to minimize you?
If you click on the one next to the double, then the one is bigger.
If I click on the what?
Like, if you click on the middle one, then you're the bigger.
Oh, that's better. Good. Thank you. Thank you, Bertie. Okay. All right. You can go check it. Check out Jordan Peterson and the lobster, his lobster hypothesis. I'm going to post that. I'm going to post that. Not for the, it won't be for the exam, but we can talk about that. Should have you guys watch that before we got together today. But I was wrestling with my computer. So here's what they did. They deleted tryptophan hydroxylase in neurons. So they have these mice. They genetically deleted tryptophan hydroxylase. What's that going to do? Tryptophan, that's up here.
You won't have any serotonin.
No serotonin. Gone. The neurons are still there, but they just don't make anything. Okay. So now in this test, here we've got some data over here. Okay. There's our wild type. So these animals would have the ability to make serotonin. The red bars are the ones that are not able to make serotonin. So what they do is they take the two animals, they have a control animal that's in there sort of like, it doesn't know this animal, and then they put either the wild type or the knockout in there, and they see how long it takes for it to attack the other animal, and then how many times it attacks them. So, and what we're going to do is we're going to, well, I want you guys to tell me, first of all, which animals are more aggressive, the wild types or the knockouts, and why? Joey.
The knockouts have, are more aggressive, they have an increased latency, or decreased latency to attack, and an increased number of attacks.
They're more aggressive. So I take away the serotonin, the animals are more aggressive. Now here, what have I done? Okay, what is 5-HT3? Consider these animals to be animals that have, these are knockouts, all right? We're going to call these animals knockouts. So what they did was, this is really cool, they then gave the animals 5, how did she end up over, oh, she wasn't, 5-HTP. That's the product of the first reaction, you know. That's the product of this first reaction, see that? So this is the enzyme that was knocked out. And now they gave them this. So they have this enzyme. So the animal can now make, guess what it can now make? Serotonin. Isn't that cool? See how scientists think about this stuff, experimenters. And what do they do? What do they find? What happens to the latency to attack now? It goes back up to like wild type levels, and the number of attacks that they make go back down. Ooh, isn't that groovy? That's nice in mice. What about humans? What do our human studies show? Here's some human data. So what is fluoxetine?
I have a question.
Yeah.
Why does serotonin, like the lack of serotonin, make you aggressive?
Why does lack of serotonin make you aggressive? Probably because, well, this is the balance of, it's like the norepinephrine story.
Okay.
So if you have none, you can be very aggressive. If you don't have any, or no, if you have none, you can be very aggressive. If you have too much, then perhaps you won't care about anything. Oh, that's nice. Not enough. Okay, so it's all like a balance. So probably not enough. You need some. I have to look. Let me look into that. Let me get an. That's a good question. I'm going to try to get an answer for that for you next week. What about humans? So this is also an anxiety, or this is an aggression score. So they're asking the people, how aggressive do you feel now? What is fluoxetine? It is a selective serotonin reuptake inhibitor, which means it's blocking the serotonin transporters. So what's that going to do to serotonin in the brain, in the synapse? By blocking transport?
Inhibit?
It's going to allow the serotonin to sit in the space between the neurons and not get reabsorbed back in.
So then what's that do, in theory, to the serotonin levels in the synapses then?
Keeps it up.
Keeps them up. You got that, Allison? A block transport, not vesicular transport. So we could say then that the red line people, after a number of weeks, their serotonin levels in their brains are going to be going up. And what happens to their aggression scores? They are lower. Something happened at week 2. Everybody went down. Maybe they got paid on this day, I don't know. Here's your check for being in the study. Thank you. Oh, I feel better. But what happened over time? Those with presumably higher levels of serotonin in their brains, their aggression, or at least their perceived sense of being aggressive, what happened to it? It decreased. And hopefully Well, it's sort of a mishmash. Yeah, we won't worry about serotonin and dopamine. So, what are some of the big takeaways here? Okay, so test. Test on Thursday. What the heck's the test going to be on? I'm going to be honest with you, I don't know yet. Look how Percy's laughing.
You haven't made it yet.
No. So, you should definitely, there's gonna be a wide variety of questions, different kinds of questions. Everything range, I mean, I'm gonna ask you questions about the brain regions. We've talked about those a lot in the first, what neurons are found there, what, in general, what functionality do those brain regions help regulate? We definitely talked about these neuronal pathways, right? We talked about the major neuronal pathways so far, the dopamine ones, the substantia nigra, the mesolimbic, the mesocortical. We talked about the locus coeruleus. Now we've talked about the rate thing. So you need to know what neurotransmitters are associated with those. Know the pathways, know the major enzymes. I highlighted to you 3 major enzymes that are important. Receptors, different types, right? Ionotropic, metabotropic, what does that mean? What about IPSP, EPSPs? What effect is that going to have on a neuron? Synapses, using different drugs to block neurotransmitter release or synthesis or breakdown, et cetera, et cetera. So there's going to be, some of it will just be sort of basic information that we've talked about. Others, questions will be, here's a graph, here's some information. Tell me what it means. So we've done some of that. We've looked at a number of graphs now. We've interpreted a number of graphs. And I'm expecting you guys to start thinking about that. All right?
I have a quick question.
Hey,
So would you like, for example, give us a drug like ritanserin and ask us what it does?
no. I'm going to tell you what it does.
Okay, and then
I'm not going to hold you to like apomorphine, go. I can't even remember what it does. How can I expect you guys to?
okay.
I'm going to give you specifics about it, but then I'm going to ask you, for example, drug Scooby-Dooby-Doo leads to, I'm going to give you a quick example here. Scooby-Dooby-Doo, 143, leads to activation of dopamine neurons that terminate in the striatum. What's something you should expect to see in those animals? What's a side? We talked about this effect gazillion times. Activating dopamine neurons in the striatum. I know we've covered this. Do you remember? Bertie's like, I can't remember.
No.
Okay, maybe that's a bad question.
Would it be lack of spontaneous movements?
Substantia nigra, hit the substantia nigra, go. What's substantia nigra regulate? Movement. What if it activates mesolimbic? Emotions.
Emotion.
Or, also talked about,
Goal-directed behavior.
I like it all, rewarding behavior, good. Yeah, so some questions like that.
So what about if there was a question that said about like smooth motor movements, would you want us to put cerebellum, or would you want us to put the
Substantia nigra?
substantia nigra?
It probably, if you put them both down, so both of those are involved. So, yeah, they're both involved.
Okay.
That would probably be more of a multiple choice kind of thing. So, yeah. Don't stress. Don't let go adrenergic, all adrenergic on me. It's going to be okay. This is not supposed to be a deal breaker. It's going to be challenging, but it's going to be okay. It's going to be okay. Well, it's not going to be easy. Who wants to accomplish anything easy? Julie did. She's like, I don't want that. It's going to be fine. You guys are going to be fine.
I can tell myself that as I'm thinking.
I'll be able to take it in 5 minutes or less.
So what classroom is that? Because I'll just come to class on Thursday to take it.
Okay, it's an E104.
E10.
E104. Yep, E104 and Dupre.
Okay, yeah, because I have a biopsych exam directly after, so
Oh, man.
yeah.
Well, then you should be ready to go then. I've heard you guys are talking about these drug effects in that class too.
Yeah, a lot of the material overlaps.
Yeah, I'm gonna be pharmacology now.
See you Thursday. Thank you.
Thank you, Father.
So is it exactly the same class?
No.
But like the class that, like what he does, it kind of like goes into like the basics of what we're doing, and then we go into more depth and like connect them, whereas they're kind of like basics, so we can like understand, psychology.
Okay, okay.
Yeah, like he talks about the brain, like the distinction between the brain and the mind, and he talks about the understanding of the brain from like 4 different psychological perspectives. So it's more psychology based.
Okay, and this is just a lot more biology based?
Yeah.
Okay, I just want to make sure I'm not just you guys are getting the same class twice.
Oh no, like we've never gone into any of this, like the pathways in great detail. We've never talked, like we haven't done as much anatomy and physiology in that class at all.
Okay.
So it is, I mean, there's big differences. In fact, I think this one's definitely more in depth.
Okay. Well, I mean, you can't do everything in your class, so he does have to get into the psychology of it.
Right.
That's important. Yeah, so you know what? You sent me, I'm going to stop